All right, so as a reminder, this is being recorded, and this is part of a webinar series that we have. So if there are any other webinars that you're interested in, please make sure to check those out. The webinar series is called Taxonomies A to Z, and the series will also be recorded and put online on our YouTube channel. And we're happy to take any questions. So here on Zoom, if you have any questions, you can please post them. And if you're watching a recording of this, please post the questions in our community forum. And so for today's agenda, I will start by introducing this webinar challenge, and then Sam will present his winning submission, and then we'll have some moments for some Q&A. And so now I will introduce the MapMySpikes data challenge. And so to give a brief background on this data challenge and who we are, I'll just talk a few minutes about the Allen Institute. So the Allen Institute is a nonprofit biomedical research center. We're about 20 years old, founded originally by Paul Allen. And over the last 20 years, there have been a number of large data sets and atlases that have been produced and released by the Allen Institute. And one of our most recent atlases is called the Allen Brain Cell Atlas, or ABC Atlas. But some of you might know us from our mouse reference brain atlas, which was released almost 20 years ago. And all of these atlases are put online on brain-map.org, all of our brain science research, because one of the values of the Allen Institute is open science. So we believe in sharing our science openly for others around the world to use. And in spirit of open science, we also believe in doing data challenges and getting others to interact with the data that we have produced. And so that's sort of spirit of this data challenge that we had called MapMySpikes. And the way that the challenge was set up was it was centered around this question of how do firing properties of a cell relate to its molecular cell type? And the request for the challenge was, we are seeking tools that can accurately make this connection and relate it to existing knowledge of cell types on Allen Brain Map. And so we know that neuroscientists have been using patch-seq electrophysiology to characterize cell types for many decades. And now, more recently, we have other cell types that are defined by single-cell transcriptomic data. And so the question is, how can we connect these two different modalities and come up with cell types that link these two different ways of characterizing them? And one way of doing that is through a technique called patch-seq, where you're able to patch from a cell, record it, and then extract the nucleus and get sequencing data from it. And so those are some experiments that we do at the Allen Institute. And the spirit of this challenge was, how do we collect and connect external patch data to this patch-seq data at the Allen Institute so that we can try to infer some of these transcriptomic identities to electrophysiological recorded cells. And so the main parts of the challenge were, one, to create an algorithm that will accurately map patch-clamped recorded neurons from 3 example data sets to the most likely cell types in the mouse primary visual cortex and or the primary motor cortex. And part 2, to present your mapping results as a user-friendly tool that links to the mapped cell types on the patch-seq VisP viewer or the CTKE, which is the Cell Type Knowledge Explorer. And so these were the 2 different parts to the challenge. Part 1 was to do the mapping, create an algorithm. Part 2 was to create a user-friendly tool to view this mapping and to see these connections to patch-seq data sets on the Allen Institute website. And so here's an infographic describing this. So I mentioned at the beginning that the whole idea of this was to map these mystery input data sets. And so what was in these data sets? So the input data sets were 3 different data sets from published papers that had been published in the last 2 to 3 years. And these were all papers that used patch clamp electrophysiology along with other types, other techniques, laboratory techniques, transgenic mouse lines, immunohistochemistry, to then map their cell types onto patch-seq data sets from the Allen Institute. And these were the parameters that were used for these 3 different papers. And so for the challenge, we removed the name of the papers. We de-identified these data sets and stripped them of any identifiers, and then gave these 3 data sets to our data challenge participants. And we only gave them the electrophysiology data. So I mentioned that, these experiments involved other ways to characterize these cells, but in this challenge, we were just focused on the electrophysiology data. And so what was provided to the participants was some information about the individual cellular recording. So here's an example of the data that was collected and distributed for our data set number 3, query 3. So what area of the brain this was from. So these recordings were all done from cortical areas, and other values from the cell that were recorded, such as resting membrane potential, input resistance, SAG, other parameters related to the action potentials. And so we gave our participants these 3 different data sets featuring just these charts, featuring just the electrophysiological data. And the goal was, can they take this data, come up with an algorithm, and map it onto Allen Institute patch-seq data sets, and then infer transcriptomic identities based on that patch-seq data. And so the data sets that we're being mapped onto are 2 different data sets that are available freely from the Allen Institute on brain-map.org. The first data set that was being mapped onto features patch-seq data from the motor cortex, primary motor cortex of mouse. And so one way to access this data is through going to the Cell Type Knowledge Explorer, which is an online tool and portal that we have on brain-map.org to access this data set. And in this data set, in this taxonomy, we have both excitatory and inhibitory cells that were recorded from and characterized using single-cell transcriptomics and also patch-seq. The second data set that we asked our participants to map onto features data from the primary visual cortex of mouse. And this was primarily inhibitory cells. And these data can be accessed online through our visual cortex patch-seq viewer, which is also on brain-map.org. And I just want to reiterate that the original 3 mystery data sets that we had, the authors of all these papers did this mapping previously in their paper to these data sets, but we removed that and de-identified them and just gave the electrophysiology data to our participants to see if they could also do the mapping purely just based on the electrophysiological data provided. And what did we do as part of the data challenge at the Institute? So we were also in charge of judging submissions. So judging the algorithms created, how accurately they were able to do the mapping, how accurate they were to the original authors of the mystery papers, and if they created a tool, how user-friendly was that tool? And these were 2 different parts. So participants could either submit just their mapping results, or they could submit a tool as well. And some people submitted one or the other, some submitted both, and both parts 1 and 2 were judged independently. How accurate were you able to map these data sets? And then if you created a tool, how user-friendly is it? And we'll be hearing today from Sam Mestern from Western University of Ontario. And Sam is a PhD student who won both of the categories. He won both the mapping accuracy, and his tool was also the most user-friendly. And we'll hear from him in the next minute or so about his submission and his experience with this data challenge. And when we finished the data challenge, we released the identities of these 3 mystery data sets, which featured the patch-clep data. And so these are the 3 different data sets that were used in the experiments for this data challenge. And if you want to read more about this challenge, read more about Sam's tool, or also read about the other submissions, From other participants, we have that information available online on our event page for this data challenge, which is at alleninstitute.org/events/MapMySpikes. And now I also want to mention this was a challenge that involved many people organizing from the conception of the idea to the execution. And so I personally also want to thank Nathan Gowans, Brian Lee, Brian Kalmbach, and Rachel Daly for their insights and help. These are scientists who are involved in the patch-seq pipeline here at the Allen Institute. And this challenge was created by Jeremy Miller and myself. And we are planning a second data challenge in early 2025. And this data challenge specifically will be related to the Allen Brain Cell Atlas, or ABC Atlas, which is an online tool that features both single-cell transcriptomic data and spatial transcriptomic data from mouse and human. All right, and now I'm going to hand it off to Sam.
Cool. Let me just share. Okay. All right. Thank you so much, Rachel. So today I'm really excited to talk to you guys and to kind of geek out about intracellular electrophysiology a bit. Today I'm going to talk about the technique I use for the MapMySpike challenge. It's a domain adaptation technique, which essentially aims to merge two distinct data sets in a way that sort of maintains structure in the data sets. And we're going to use this on our intracellular electrophysiology to get some new insights and hopefully find some interesting things. So the overview for my section of the talk, today I'm mostly going to talk about the motivations for the project. I'll talk a bit about the method, and then I'll show you how we're using it here at University of Western Ontario to get some novel insights from our data we've gathered. So talking about the motivations, I want to first say that I am actually, my day job is a patch clamp electrophysiologist. There's me sitting happily in front of my rig. I study under Dr. Wataru Inoue, and through close collaboration with Dr. Julio Martinez Trujillo, we've been able to record from NHP cortical cells here using patch clamp electrophysiology. Here is an example of me patching a cell. This is supposed to be a video of me trying to suck the nucleus up, but the video is not working. And here's an example electrophysiological waveform of one of the cells. Now, we've been actually working on this project since 2019, and we've gathered quite a large database of cells with really distinct phenotypes. And we really wanted to sort of derive novel insights from them, and we also wanted to generate biologically constrained models of our subtypes. However, we quickly ran into a problem when we started this project, which is we ourselves didn't have any sort of cell type information for our NHP cells, since at the time we didn't really have any viral tools, nor do we have transgenic animals, and we weren't able to perform patch-seq at the time. So we were sort of left wondering what the different cell types of our neurons are, and we thought we might be able to derive that using the ePhys features. And this is sort of what led us down the path to use the Allen Institute's open data sets with, specifically in this case, the mouse data sets, which have those labels, and apply them to our NHP data. And we really first off wanted to merge the mouse, human, and our NHP data to get this sort of comprehensive map of cell types. And that's sort of where our thinking led us, and this sort of question kind of meshed well with the MMS data challenge. So one major problem with merging a bunch of ePhys data is that the intracellular electrophysiology data can vary greatly with experimental conditions. This includes things like temperature and setup and solutions, and it may not even be visible to the naked eye. Here I pulled two cells from one set of cells from the Tobias lab or the Scala et al. Dataset, and one set of cells from the Allen Institute. Here is a pyramidal cell from both labs, what look quite similar to me. Here's an SST cell from both labs. Again, looks quite similar to the naked eye. However, if you were to visualize these data sets on a sort of like whole broad spectrum, you can see that overall the Tobias lab dataset is slightly shifted compared to the Allen Institute dataset. And that's likely a result of different recording conditions, which can greatly affect the extracellular or intracellular ePhys features that you're recording from. In this case, I believe it's actually the Tobias lab dataset I have here was recorded at a different temperature from the Allen Institute dataset, which causes this sort of feature skew. So that led us to our problem, and now we're going to talk about the solution, which is the method I worked on for the MapMySpikes challenge. So the method is actually twofold. Whoop, those popped up in the wrong order, but step one is to use domain adaptation to integrate the data sets, and I'll go a bit into that in a minute. And the second step is to use hierarchical classification to assign the cell types. So domain adaptation is sort of a fancy machine learning term that talks about a technique that aims to bridge between dataset drift. And what do I mean by drift? We're going to use a simple example here. Let's say you're a machine learning scientist and you make a classifier to classify photos of ducks and photos of swans. You make your classifier, it works quite well. However, you want to now apply your classifier to photos of ducks and swans, but now taken at night. The underlying distribution and sampling is the same. There's still photos of ducks and swans, but the context of the data has changed, and this has caused dataset drift, meaning it doesn't occupy a similar feature space as your original data. Uh, Domain adaptation aims to take that data and sort of place it back onto the original feature space and allow you to use your original classifier on that data set. And my underlying assumption here is that the underlying distributions of the cell types is similar. However, the context in which those cells are being recorded from is changing. Similar to the duck photos shifting from day to night, you're recording from like a pyramidal cell, either in a low temperature or room temperature or in body temperature. So to perform domain adaptation, I chose to use an optimal transport formulation. There's also other proposed methods, but I preferred optimal transport because as a non-math trained individual, I found it quite intuitive. Optimal transport defines a whole field of mathematics, but the general idea here is that it's measuring the distance between two distributions, and it's more formulated as the sort of cost to transform one distribution into the other distribution. It was originally thought to be devised by a mathematician who was looking at transforming one dirt pile into another dirt pile, hence the sort of drawing of the stick figure here. What it looks like in practice is something like this, where you have two distributions and you create something called an optimal transport plan, which tells you the cost of turning one distribution into the other distribution. And what you can do with this plan is actually trigger that transformation. And so what this looks like in practice is something like this, where you have two samples, a set of source samples, a set of target samples. You compute your transportation map, or the lowest cost to transform one distribution into the other, and you apply that transformation, sort of moving the one domain into the other domain. In this case, we're also going to use a regularization term called Laplacian regularization, which simply constrains based on the internal graph of the data set. And we can talk more about this in the question period, but I won't go too far into this. Okay, so I first wanted to, oh yes, to perform this technique, I'm going to be using the Python optimal transport package. And for the MapMySpikes challenge, I submitted my package, the patch ODTA, but it really just wraps the Python optimal transport package. So a lot of the credit for this work goes to the POT maintainers here. Patch ODTA just simply introduces a wrapper for easy switching between the different optimal transport techniques and a couple of other features that I found to be useful. Okay, so I'm first going to test this technique by working with a data set I'm going to call the Gowans 19 data set. I'm not sure what it's referred to initially, but this is the Gowans 2019 paper data set, which features the patch clamp data labeled by a Cree line, not labeled by transcriptomics, as well as the CTKE data set, and I'm going to merge them into one data set. Now, if I showed you before, the distribution between these two data sets the distributions look similar, but they are skewed slightly. If you were to perform simple z-score normalization, it does appear to work. At least the CTKE dataset does seem to overlap with the Gowans 19 dataset. And furthermore, if you perform OTDA normalization, we see a much better mapping of that CTKE intermingling with the Gowans 2019 dataset. And this is much more visible on a UMAP plot, where if you were to UMAP embed the sort of z-score dataset, you see that each individual dataset is forming its own little island in the UMAP, which is likely meaning there's some sort of feature in there that's causing the UMAP to pull the datasets apart. However, if you were to UMap embed the ODTA merged datasets, we do see a good intermingling of the two datasets. I then, as a sanity check, wanted to make sure things were sort of where they should be. So I looked at the labels given by the Cree line and the Gowans 19 dataset, and the labels given by the transcriptomics in the CTKE dataset. And you can see that the PV labels and the SST labels and the VIP labels for both datasets on the same UMAP projection do overlap quite well. That's again reflected over here, where I've drawn just big circles around the CTKE dataset and the Gowans 19 label in the scatter plot. And you can see again, the PV, SST, and VIP do overlap quite well. So this was quite encouraging. But, oh yes, if you train a classifier simply on these merged datasets, you can see the ODTA improves the sort of transference of the classifier between the two label spaces, getting an accuracy of about 0.7 compared to an accuracy of above 0.5 for the Z-score dataset. A final sanity check is I looked at the ephys waveforms of cells from the CTKE dataset and the Gowans 19 dataset, with the CTKE dataset being in red and Gowan 19 being in black here. And you can see the ephys waveforms do seem to match with their nearest neighbors. Here we have, I think, a PV cell from the Gowans 19 dataset, which matches quite well. Here is a collection of VIP or SST cells, which again match quite well. A pyramidal cell and probably another pyramidal cell here on the right. Okay, with that in mind, I wanted to talk a bit about what happens if you have really imbalanced data sets. This is something that was the case in the MapMySpikes challenge. And to simulate this, I took the Gowans 19 dataset in full, and then I took the CTKE dataset and subsampled it down to mostly PV cells and 10% other random cells. The idea here is that it's simulating what happens if you were maybe patching Cree-targeted PV cells, but you also accidentally got one or two. If you were to simply map them on using standard ODTA, you would see that the Telias PV cells begin to intermingle across the entire spectrum of the Allen dataset, which is not good. It's meaning the PV cells are invading the sort of pyramidal SST and VIP space. However, if you use the regularized ODTA or the semi-supervised ODTA, we do see a sort of constrained spread of the PV cells, meaning that now the CTKE cells are sort of remaining in the space where you expect them to and are intermingling with the same cells from the Gowans 19 dataset. This is to say you should be probably using a regularized ODTA and the semi-supervised ODTA when possible, since normal ODTA kind of over-integrates the data sets. Okay, so let's talk about the MapMySpikes challenge. Here, as Rachel presented, we had several different queries, and we are given some amount of information on them. And using that information, I chose to map differently for each query. In this case, query one, I knew at least it was likely SST cells, and I chose to map it against the complete VISP dataset. Here is the UMAP embedding, with the reference data being in the smaller dots and the user data being the query data being in the bigger dots, and the sort of transgenically driven labels here. And you can see that overall, we do see a good mapping of the query data into the SST space. For query 2, I wanted to map it since we know it's layer 5, but we know it's probably encompassing both interneurons and pyramidal cells. I wanted to map it against the CTKE dataset. And again, we do see good spread of the query 2 data amongst this dataset. And finally, for query 3, I knew it was SST again, so I mapped it against the VISP dataset, or V1SP dataset. And again, we see good mix amongst the SST data there. Okay, so now we have our integrated data, and we're going to talk about how I actually generated these labels. So my second step here was a hierarchy classification to identify cell types. And when I'm talking about hierarchical, it's actually a hierarchy of hierarchical classifiers. So what we're using here is a, oh, I'm missing a slide. That's okay, but what I'm using here is essentially a forest of forests. So it's a random forest at 3 different levels, which is then predicting the subtype layer for each level. So level 1 represented our overarching subtypes. So these were representing our sort of major subtypes like LAMP5, PV, SST, and VIP, and overall level 1 random forest functioned well. From here, once level 1 predicted its label for a cell, we moved down to level 2, and this forest's job was to predict the subtype of each cell. So level 1 would predict something like SST. Level 2's job was to predict what subtype of SST it was. Here, I am a little bit underwhelmed by the results, but I do get an overall test accuracy of 48%, which is good given the amount of labels it actually needs to predict. And you can see amongst the diagonal here, we get somewhat good accuracy of the subtypes. Moreover, the cells that are sort of grouped within their subtype are grouped appropriately, meaning the PV cells are being misclassified as other subtypes of PV cells and so on. And this is further seen at level 3, where we move down to that third level of label. And we do see a good sort of representation amongst the diagonal on the confusion matrix here. But overall, it's a little bit underwhelming, that accuracy. So I'm totally open to any suggestions about improving the classification accuracy at level 3 here. Okay, with that in mind, I wanted to talk about the results I got. For query 1, overall, I got fairly good prediction results where the SST cells were being predicted as SST cells, with the true labels being on the y-axis and the predicted labels being on the x-axis. However, I did see a bit of intermingling amongst the SST subtypes, meaning the classifier was not getting the SST subtype exactly right. Similar idea here for query 2, where I did see good overall general classification, meaning the pyramidal cells were being classified as some type of pyramidal cell, the PV cells as some type of PV cell, SST is some type of SST, and so on. However, the exact subtypes, again, were not predicted well. And finally, that's same case for query 3, where we saw good prediction of SST subtype, or good prediction of SST overall, but sort of lower accuracy when it came to the specific subtype of SST we were predicting. So with that in mind, I've shown you how we use the main adaptation to integrate the data sets. And then I've talked about how we're going to use, or how I use this classification method to sort of classify from a major cell group down to the sort of very specific transcriptomic subtype. And now I'm going to move on to talk about really quickly some applications we've been using here at UWO. So at the beginning of the talk, I talked about how we wanted to use the Allen Institute's mouse and human data sets to label our NHP data sets and possibly derive some novel insights. To do so, I took all 3 data sets and ran them through IPFX-based feature extraction. This is actually a package put out by Allen Institute, which extracts features from electrophysiological waveforms. So I ran it across all 3. I got about 146 ephys features, and then I performed the domain adaptation. Here you can see the input resistance to Rio-based action potential width curves for the mouse dataset coming from the Allen Institute, and the NHP dataset coming from us here at UWO. And you can see they do somewhat integrate quite well, although we have a couple of outliers here at UWO. If you were to perform z-score based normalization, interestingly, we see somewhat of a cluster of NHP cells at the sort of center of the mouse data, which is meaning that the z-score based normalization isn't functioning quite well. However, if we perform ODTA normalization, we do get a good intermingling of the NHP and mouse subtypes. This is further exemplified if we look at the UMAP, where when we embed all 3 just by simply z-scoring, we see sort of clusters of species on the UMAP, whereas if we use the ODTA-based method, we see an intermingling of the mouse, human, and NHP cell types. Finally, we did take a little bit of a look at the mouse labeling, and we wondered what would happen if we were to transfer that mouse labeling to our NHP data. So we did so here. Here I have the mouse transgenic cell types labeled in the sort of transparent, and I've labeled our NHP data based on its nearest neighbor in the data set, based on its nearest mouse neighbor in the data set with a sort of solid color. And you can see we do see good label transference here, and the sort of PV cells are where the PV cells are, and the pyramidal, and so on. Finally, we did a check to make sure, like we did with the Telias dataset, that the datasets were integrating well by looking at the ephys waveforms. Here I pulled out one of our PV cells, And it does look quite similar to the PV cells, the nearest neighbors from the Allen mouse data set. Again, here's a pyramidal cell. It looks quite similar to the pyramidal cell, its nearest neighbor from the mouse data set. And finally, down here is a VIP cell, which does look quite similar to its nearest neighbor from the Allen mouse data set. So with this in mind, we actually wanted to see if we could get some interesting insights from merging all this data together. To do so, I split the data into the excitatory and interneuron clusters, and then I performed Gaussian mixture model clustering, similar to how it was done in Gowan's 2019. And then I wanted to look at the eFIS features from the unnormalized data based on these classifiers or this clustering we did with the normalized data. And so I pulled out a couple of interesting observations here. Here, looking at SST cluster 2 in the sort of light green, you can see that the sag in this cluster is higher in the NHP compared to the mouse and human, but the human also having a slightly higher sag than the mouse. But the first ISI doesn't differ. A similar story for PV cluster number 2, which is this light red right here. And then if you look over here for pyramidal cluster 1, which is on the far right, we saw an increased sag in the NHP and an increased sag in the human compared to the mouse, and an increased sag in the NHP compared to the human. And most interestingly, we actually saw a lower first ISI in the NHP compared to both the mouse and the human, with no difference in the first ISI between the human and mouse in this cluster. And then the very final thing I'll mention is we were originally building these labels so we could build biologically constrained models of cortical circuits in silico. And now that we actually have these labels, we're now able to build a sort of biologically constrained model. So here is a pyramidal cell that I've labeled using this domain adaptation technique and its respective model, similar for PV and similar for SST. And this is sort of a future directions where we're hoping to take the labeling. Okay, so overall, I showed you how we could take our data, merge it with the Allen Institute, and provide labels. And I also showed you that there's possibly specific subpopulations of cell types with divergent properties. And then overall in the talk, I talked about my motivations for this project, how the method works, and then some applications. And I just want to do a quick shout out. I have some other work that people may be interested in, one of which is the primate database, which is a sort of Allen Institute-like web interface for our data we've gathered across the NHP project here at Western. And you're welcome to come check it out. I worked on the back end on this, so I'm very proud of what I did. And second, if you're a big intracellular electrophysiology geek like me, you can check out this Intra-Dandy Explorer, where I took a bunch of intracellular open data sets on Dandy and kind of tried to feature extract them and merge them together, and then also made a little sort of database visualizer. So you're welcome to check that out if you're a nerd like me. And that's everything. I just want to thank Wataru and my fellow patchers, Julia and Michael and Michelle, and everyone at Neurodex. And feel free to email me if you have any questions. Thank you.
Okay, thank you, Sam, for explaining your method and telling us about some other really interesting projects that you have going on that are very highly related to the challenge. It seems like it was a good fit for you. I guess we can wait a minute or two for questions. And if there aren't any questions in this live audience, I have a few more slides that I can share as well, just to wrap things up. So I do just want to highlight some other resources that we have at the Allen Institute for those who are interested in studying cell types, whether from an electrophysiological standpoint, from a transcriptomic standpoint, from a patch-seq standpoint, morphological standpoint. We have resources to address, some of those questions about those data sets, our protocols, if anyone has any questions, specifically about how certain things are carried out at the Institute. We have a number of guides, tutorials, white papers, available webinars, use cases, data sets, portals that are all relevant to today's topic. And we also have a community forum where if you have particular questions about certain data sets, how things were carried out, understanding certain data portals on the website, we have, I think, 4 or 5 years of history of community posts on this forum where people ask all sorts of questions and our scientists come and answer. And so you have a particular question, there's a possibility that it's already been asked and answered on this community forum and you can read about it. Or someone else might have the same question as you. And by you asking a question on the forum, you might also be helping out someone else. So these are 2 resources that I just wanted to highlight. And for those watching, we also have, again, this is part of a webinar series. Today is November 20th. This is webinar number 2 in our year of these webinar series. We have a few other webinars coming up. Our next one will be in December, which will feature a Shiny app that was created for comparing annotations in annotations of transcriptomic data of cell types across different studies. In January, we'll be having a talk focused on the nuances between single cell and single nucleus transcriptomics. In February, we'll have a webinar from members of our Seattle Alzheimer's Disease Brain Cell Atlas research team. And in March, for those who are good at programming and are comfortable using Python, we'll show how to use a number of Jupyter notebooks that we have to access the single cell and spatial transcriptomic data that we have featured in our ABC Atlas. And we also have a workshop coming up at the Allen Institute in March. This is primarily geared towards graduate students and postdocs, and the submissions are due on December 6th, and this workshop is centered around cell types and how do we characterize them, what are the different methods we use, what data sets exist to help further your research, and we are providing a $1,500 travel stipend for that workshop as well. All right, and I don't see any other questions in the chat, so I think we'll end the webinar a little bit early. I want to say thank you again, Sam, for participating in this challenge, and for everyone else who submitted to this challenge. We had people submit from a number of different countries, so it was really exciting to see people from all over who were excited by this challenge and who were engaged and who spent a lot of time tackling a very difficult problem, a very difficult data challenge. So thank you to everyone who submitted. We had a great time, looking over all the submissions and seeing the great work that everyone did. And please stay tuned for early 2025 for our next data challenge that will be featured related to the data featured in the Allen Brain Cell Atlas.